Okay, so welcome to this next video in the playlist on calcium signaling. In this video, what we're going to do is we're going to look at how we can use electrical oscillations, i.e. action potentials happening across a membrane to produce calcium oscillations. So we're going to study electrically triggered calcium oscillations, and in this video we're specifically going to look at this in the neuron. So electrically triggered calcium oscillations is what we're going to look at. So we've looked now at calcium waves and how we can trigger those through the IP3 pathway, but there are other ways of triggering an oscillatory calcium concentration in a cell, and we're going to look at one now: electrically triggered calcium oscillations. Right, okay, so let's say we have a neuron, and I'll draw a neuron once I've underlined the title. Right, okay, so let's say we've got a neuron here. So these are the dendrites of this neuron here. Then it's got its cell body. Let me just draw some more dendrites. So these are dendrites spanning out, and they'll have synaptic contacts with other neurons. And then here is the cell body at the center here. So this is the cell body, or the cell soma. And then here is the axon going off like this. And then it will have an axon terminal over here somewhere. Right, okay, so if this neuron fires action potentials at a certain frequency, so let's say it fires 3 a second, so in a second it's going to fire 1, 2, 3. 1, 2, 3. So it will continually fire action potentials down its axon. Then if we look at what's happening in the axon terminal will be continually receiving action potentials. So let's just have a quick reminder of what happens in an action potential. So let's take out a little piece of membrane from this axon terminal over here. So here's a little piece of membrane from this axon terminal. And let's have a look at what's going to be happening across that membrane. Okay, so from the axon, what will happen is when this neighboring portion of membrane to the axon terminal, let's say this neighboring portion here in orange, undergoes an action potential, what will happen is that the electrical potential difference across this orange piece of membrane will depolarize, and it may reach highs of maybe plus 40 millivolts, okay, which means that the intracellular compartment has an electrical potential 40 millivolts higher than the electrical potential of the extracellular compartment. So what actually moves in order to produce that change in electrical potential? Well, it's sodium ions. You move sodium from the extracellular compartment to the intracellular compartment, and that raises the electrical potential of the intracellular compartment and reduces the electrical potential of the extracellular compartment, and therefore depolarizes the electrical potential difference across the membrane. So you have moved in some additional sodium that has carried this positive charge into the intracellular compartment just below this orange piece of membrane. And now what's going to happen is that some of this sodium is going to diffuse over into the cytoplasm of the neighboring membrane. So what's going to start this all off is that this pink piece of membrane is going to receive some sodium from this neighboring cytoplasm over here. And basically this sodium has a positive charge, so it is going to make this cytoplasm slightly more positively charged, i.e. it's going to raise the electrical potential of the intracellular compartment underneath this pink piece of membrane. Now, if you raise the electrical potential of the intracellular compartment underneath this pink piece of membrane, then if we look at the electrical potential difference from extracellular to intracellular, which means if you had a little man sitting in the extracellular compartment measuring electrical potential, and he moves into the axon terminal and measures electrical potential in there, what's the change that he sees? Well, originally at resting membrane potential, it's around negative 65, i.e. he would go down by 65 millivolts moving into the intracellular compartment. However, if you're raising the electrical potential of the intracellular compartment, then this number is going to depolarize, basically. So, you're going to depolarize the electrical potential across the cell membrane. Okay, so let's draw a graph over here. Alright, and what we will do is we will plot the voltage across this pink piece of membrane from extracellular to intracellular against time. So it starts off at this resting membrane potential, which is around negative 65 millivolts in most cells. Okay, now when this sodium diffuses in here, that causes the electrical potential difference across the membrane to depolarize, i.e. become less negative. Now, if the electrical potential difference across the cell membrane goes above a certain threshold potential, which is usually around negative 40 millivolts, then what it will cause to happen is it will cause voltage-gated sodium channels in the cell membrane to open. So let's draw a voltage-gated sodium channel here. So voltage-gated sodium channels consist of 4 domains, basically. At least the pore-forming unit consists of 4 domains. So you can divide it up into 4 domains, domain 1, 2, 3, and 4. However, it's made by a single polypeptide. So a single gene codes for a single polypeptide, which makes this entire pore-forming unit. And there's the pore going down the middle there. However, you do divide it into these 4 domains, which are very, very similar in structure. 1, 2, 3, and 4 here. Okay, right. This is what's known as the alpha subunit of the voltage-gated sodium channel, and the alpha subunit forms a perfectly feasible channel on its own. However, it's often accompanied by auxiliary subunits, which are known as the beta subunit. So here is a beta subunit. So there's our voltage-gated sodium channel, and the beta subunit can alter the electrical potential difference across the membrane at which this voltage-gated sodium channel becomes active. However, the general sort of archetypal threshold potential for voltage-gated sodium channel activation is negative 40 millivolts. So, when the electrical potential difference across this membrane is depolarized to negative 40 millivolts, what will happen is this voltage-gated sodium channel will open, so it will go into the activated state. It will open, and basically what will happen is it will allow sodium cations to move through it. Now, sodium is very, very high extracellularly, and it's low intracellularly. So you've got a small concentration here, a high concentration here. So the concentration gradient is favoring the movement of sodium in, because if you've got a very high concentration of sodium ions out here, let's say, which I'll denote by these pink dots, and a low concentration of sodium in here, the chance that a sodium ion is going to hit from this side and go through is greater than the chance that a sodium ion will hit from this side and go through. So overall, the movement from extracellular to intracellular is going to be greater than the movement from intracellular to extracellular. So you get a net movement of sodium into the cell, basically. Also, you have to factor in the fact that the electrical potential difference across the membrane is still negative 40 millivolts, which means that the electrical potential of this intracellular compartment is lower than the electrical potential of the extracellular compartment, and it's lower by 40 millivolts. Sodium ions have a positive charge, which means they want to go to areas of lower electrical potential. So that basically also provides a driving force for the sodium ions to actually come into the intracellular compartment, and it's going to raise the probability that a sodium ion comes from the extracellular compartment into the intracellular compartment, and lower the probability that a sodium ion goes from the intracellular to the extracellular compartment. So it's going to overall increase the net movement of sodium through this channel. So, the result is that when you reach negative 40 millivolts and activate these voltage-gated sodium channels, you result in a large current of sodium moving into the cell. Now, sodium ions have a positive charge, so you're bringing positive charge from the extracellular compartment into the intracellular compartment. That's going to cause the electrical potential of the intracellular compartment to go up and the electrical potential of the extracellular compartment to go down, which means that the difference, the voltage difference across the membrane from extracellular to intracellular, which is the electrical potential intracellularly minus the electrical potential extracellularly, basically, if this one's going up and this one's going down, this number is going to become more positive, and indeed that's what you see. It goes rapidly up like this. and it usually manages to depolarize the membrane to around plus 40 millivolts. Now, after a certain amount of time, these voltage-gated sodium channels close, they inactivate, so they stop conducting sodium ions, okay? Now, just about at the time when the voltage-gated sodium channels close, the voltage-gated potassium channels are just about starting to open. So in your membrane you also have voltage-gated potassium channels, and these voltage-gated potassium channels are also consist of their pore-forming unit is also divided into 4, but this time the division is real. It's not just that you have a single polypeptide which has 4 similar domains in it. This time you really do build a voltage-gated potassium channel out of 4 separate proteins. Okay, so these 4 different subunits of the voltage-gated potassium channel come together and they form the pore of the channel. Now, basically, these voltage-gated potassium channels, they are usually activated at a similar threshold potential to the voltage-gated sodium channels. However, they take much longer to open. So basically, as soon as you get to negative 40 millivolts, the voltage-gated sodium channels open, and they do their stuff. They let the sodium in, and then they get fed up, so after a certain amount of time, they close, they inactivate. And at the point that these have finally sort of said, oh, we've had enough of this, the voltage-gated potassium channels have clunked along and they've just about got to the point where they're ready to open. So they start opening just at the point that the sodium channels, the voltage-gated sodium channels, start to close. Okay, so they're both triggered to open by the same thing at the same point. It's just these ones open much quicker than these ones. Now, so what are the voltage-gated potassium channels opening going to do to the electrical potential difference across the membrane? Well, potassium is much higher intracellularly, and it's low extracellularly. So concentration, the concentration gradient is favouring the movement of potassium out. Also, you're at an electrical potential difference now of plus 40 millivolts, which means that the intracellular compartment is more positive than the extracellular compartment. So the potassium ions are going to prefer to be in the extracellular compartment. So both the electrical gradient and the concentration gradient favour the movement of potassium out. So when this voltage-gated potassium channel opens, it conducts potassium out of the cell. So basically you lose the current of sodium moving into the cell and you gain the current of potassium moving out of the cell. Now, potassium carries a positive charge, so you are moving positive charge out of the intracellular compartment and into the extracellular compartment. So that overall means you're going to reduce the electrical potential of the intracellular compartment. This is going to go down, and you're going to raise the electrical potential of the extracellular compartment. This is going to go up. So this is going down, this is going up, this is going to become more negative. So what happens is that you repolarize the electrical potential across this membrane, like so, and then you go back to resting membrane potential. And that's pretty much the action potential. Okay, so how can you use this to generate an oscillatory calcium concentration in this axon terminal? Well, basically, if the neuron is continually firing, then if we plot the electrical potential difference versus time on a graph, then what we're going to see is a continuous trail of action potentials. We're going to see action potential, which corresponds to a neuron firing, and then it will go back to resting membrane potential. Then we'll see another action potential, and it will continue on. So this action potential process will oscillate basically when the neuron fires. So every time the neuron fires, you'll get an action potential down here. So you're getting oscillatory electrical activity across the membrane of this axon terminal. Right, so all we need to do is put some channel for calcium in the membrane, which will be activated when an action potential occurs. And we'll just continue this discussion in the next video.